Residues. Does anybody remember if you have guanine, it looks like this? Some nitrogens there. 6 members here. Remember, there's 5 brothers. They hate each other. This guy here flew down here, left some guanine, now he's down here. So guanine looks like this, and it's attached via this nitrogen to the ribose. So you know that guanine, does it make 1, 2, or 3 base pairs with the next base? Like when you're base pairing?
3.
You said 3, and who's his partner? Cytosine. So cytosines must have here, it must be a plus charge because that's going to be a delta negative. It must be here, and this is going to be there's an H here. So there's those 2 H's there. I should circle them. Like there's a delta plus right here, and then there's a delta minus on the green. So the reason why I took the trouble to draw that structure is when you get exposed to UV radiation, sometimes you get this. Change the color so it sticks out a little bit. That guy should not be there. You should not have an 8-oxo guanine. The reason why, this is supposed to be ribose here. So the reason why you shouldn't have an 8-oxo guanine is once you put that on there, this bond here rotates a little bit, and it causes these minus plus plus to no longer be involved in the base pairing. It causes, if you could just imagine this bond here, like spring-like, and he can just kind of bend over. So the bottom line is he gets a new partner. Instead of being partner with a C, partners with a T, A. Anyway, you get a problem. That sometimes when you get that, you get a mismatch. And we get the mismatch because maybe this is the 8-oxo up here, 8-oxo, and it doesn't want to bind with its previous partner, which previously should have been a C. So you get this bubble. Thus far, whenever we talked about damage, all we said was you gave an 8-oxo and it caused a mismatch. So these guys don't like to bind. Like originally it looked like this. There was a G and it was a C, and this region here looked like that. And everybody was happy because the G was not modified. But if you make 8-oxo guanine, now I put a little question mark because G no longer wants to bind to C. So you have this bubble. That's not a break in DNA. You could get a whole break in the DNA when you literally snap this. And I haven't actually said what causes that, and I actually don't know how. I don't know the chemical process by which you break the backbone. Like it would literally be on this is where it would happen. It would be right here. Here's your, remember this is 1, this is your 2-hydroxy, this is your 3-hydroxy, it's DNA. We don't have the 2, only RNA would have that, 3 does have it. And here's where you bind. So remember we said that when you're binding with DNA, there's basically the 5' end has a hand, and he's the guy that does the grabbing. And then here's the hand, this 3' OH, that's where you have like a little handle that you can grab a hold of. So this is your 3' OH. So when you're cutting DNA, making a permanent break or double-stranded break, you're actually cutting right here. You're cutting, that's supposed to be scissors. You're cutting right there, or here. You're actually cutting the sugar phosphate backbone. So you could cut this carbon, you could cut there's a phosphate here, going that way, and there's a phosphate this way. So when you cut that bond, you end up with this. It's a double-stranded break. And that's bad. But let's just say for argument's sake that it went like this. I'm going to draw the neighborhood around it. Let's just say it went A, and the top one was a G, and we'll say this is A, T. And the bottom strand obviously was T, C, this would be A. And just a second ago we said that was oxoguanine, so we're going to put a little star, and now these 2 bases are not getting along with each other. But if we did a double-stranded break, there'd actually be a cut, and it really wouldn't matter if the oxoguanine was there. And my instinct, I don't have proof of this, but once you have an oxoguanine, the probability of having a double-stranded break goes up. But that's not the point. The point is, once you've broken it, you have a double-stranded break, and these guys are still not getting along with each other. This C is like, I'm trying to stay as far away from this G, because we don't really base pair. This A and T are fine, everything else is fine. But now you have this double-stranded break. So, for argument's sake, let's say you're trying to fix that. If somebody cuts something, you go back there with tape and you tape it back together. Well, obviously there's a problem here. That guanine is bad. It's got 8-oxo. So you could come back with your repair enzymes, and the repair enzymes, you would hope they'd be smart enough to tell the difference between who's the bad guy. Is it the C or the G? And I told you last time, they're not that bright. Sometimes they take out the C. In which case, they take it out and say, sorry, you don't match, and what they bring in is a T, because a T will match up with 8-oxoguanine, and then that ends up giving you a mutation. But let's just say, for argument's sake, this time we get it right, and they take out this G, and he's gone, which means there's a protein in here. I'm gonna draw it as a big old green rectangle. This is a repair enzyme, DNA repair enzyme. And it comes in there, grabs the G, cuts it out, so it means he has a little pair of scissors. He cuts the DNA, tosses them out, reaches around, finds another one that's not damaged. So that must mean that DNA repair enzymes have a pair of scissors, and they can do base pairing. So he reaches over and he grabs, there's all possible letters, and he says, let me grab a fresh G. And he puts a fresh G in here. I'm gonna put it in a little box so you can see the difference. He gets rid of the damaged G, and it's still floating around. It gets destroyed later. And then he puts in the good G, and that causes this carbon to go back together. But we still have not solved this problem. We fixed the bad base, but we didn't glue the backbone together. Remember how I said we cut the backbone? We cut the covalent bond between this letter, or this A and that G, and on the other strand, the T and C. So we gotta glue that back together. That enzyme has a name. You should know the name of that enzyme. That's called DNA ligase. It's a different enzyme, so I'm gonna draw them in a different color. DNA ligase. It just makes a good old-fashioned double-stranded, single or double-stranded, does each strand, just ligates them back together. But here's the problem. What if suppose after you had it cut, before ligase could come in there, these guys broke off? So now DNA ligase is going to put, like, let's just say for argument's sake, there's a C and a G over here. So there's a whole row of C's. Like 3 C's in a row, 3 G's. So it's not supposed to look like that. There's supposed to be, wasn't there supposed to be a, did I say an A on the top? It's supposed to look, let me draw it correctly down here. Wasn't it an A, a G, and a T like that? Is that what I had written a second ago? Okay, good. So now we're going to basically connect this G to one of these C's here. And we've lost this. This is now gone. So the DNA ligase comes in there. He's not smart enough to know that you just lost these 2 red guys. And he's going to glue that back together. Why is that a problem?
So, mutation.
He thinks that could be a mutation. Are you sure that's really a mutation? Let's just check. All right, here's some sequence. ATG, what amino acid am I talking about with that? Yeah, that's methionine. Remember TGG? In RNA it would be UGG, which is UGS, and UGS cause you to trip. So what amino acid is this? That's right, it's the W? Yeah. And then let's just do another one. Does anybody know another amino acid codon offhand? Triple G is my favorite one. Because what amino acid starts with G? Glycine. Single letter glycine. Glutamine does, but glutamine's single letter abbreviation is E. But triple G is the only one that works like that. Its DNA abbreviations, or GG, whatever, it actually codes for glycine. All right, and then what about this? What amino acid is that? trick question, that's a UGA stop codon. So here's a tiny little amino acid, and in this amino acid, don't we wanna see maybe a triple C and then an AGT? Alright, well, I better squeeze that in there. Triple C, sorry about this, triple C, and then in red, I'm gonna draw AGT. AGT. So triple C is proline, and AG, I believe AG anything is I think that's arginine. Yeah, AG whatever is arginine. Yeah. So, sorry about that. Arginine is R, proline is P. There. Alright, so now we have, I'm gonna get rid of who we're supposed to get rid of. Aren't we supposed to get rid of the A? If we get rid of the A, I'm gonna put a little box around it. So do we still have methionine? Sure. Proline? What's the next amino acid? What is that amino acid? Who can tell me what that next amino acid is? You look up in your codon chart. It's an amino acid whose triplet, I'm gonna put it in a little bracket under it, is GUU or GTT. You could like ask your phone, what amino acid is GUU? Something like that. It would say valine. Isn't that right? It's valine. So this should, so we used to have an amino acid that had arginine, and whoops, we changed it to valine. Well, aren't they almost identical? What's the difference between arginine and valine? What characteristic is valine? What characteristic is arginine? Didn't one guy have charge? Which one's charged? Got a plus charge. Polar. This one here is neutral. And he's non-polar. Neutral, non-polar. So this is a big difference. Besides that, we're also going to get this amino acid. Which amino acid is that? It's good. Glycine and then another glycine. And it looks like we don't have a stop codon, so this protein is going to be too long. So we got a couple of problems. We ran the stop sign because it's out of frame. This is called a frame shift mutation. So we have a frame shift mutation. So that would be a bad thing. And a lot of times a frame shift mutation will introduce a new stop codon where you weren't supposed to, or it will run, this TGA is not, like whatever the next amino acid is, it starts with GA something, which is valine. Right? Anything that starts, sorry, GA could be either GA something, serum, yes, aspartic acid or glutamine. GA something is a negatively charged amino acid. But whatever the case may be, it's not going to be a stop codon. GA something is either an aspartic acid or a glutamic acid. Does anybody have the chart? What's GA something? I'm pretty sure it's a negatively charged amino acid. So on the just so you know, on the final exam, you will be given a codon chart, and you should be able to look at DNA and translate. You don't have to memorize the codon chart, but like on the back page, I'll put like the codon chart so you can practice doing that. So you can just look up, you know, what is triple C? Just look up. So it's whatever it is, it ends up being negative, right? So here's what would happen if suppose we came in here and we didn't just lose the A and the T, we lost the A, T, the GC, and the T. We lost this whole package. Gone. Lost all three. So we did the break and we lost two on the right side and one on the left. So the break is still occurring. This is where the break is, but instead we're killing that amino acid pair, well, not a pair, this base pair and these two. So what would that do to our protein sequence?
We just lose the arginine.
Yeah, don't we lose that, but we still have proline? and then after proline, we still have the TGG. So that's an in frame, that's a deletion mutation. So if you were to lose all three, that would be a deletion mutation. So we lost a nice, neat codon. We didn't get out of whack. So this is a deletion. And sometimes you would put in parentheses in frame. So we're staying in frame, which means the triplets before and after are not, well, particularly after, are not affected. So you could have insertions that cause problems, but if you're gonna add stuff, make sure you add 3, maybe stay in frame, or if you're gonna delete 3 or multiples of 3. So here's the problem. Let's say we lost all 3. And this is a in-frame deletion, and we're going to be missing this plus charge amino acid, which could be a problem. Let's just say, for argument's sake, this protein really likes having a plus charge. TGG right after the break, yeah. So here's the problem. See if you can answer this question. This is the thinking question. If this is broken and you have repair enzymes, and they come in and they do their work and they miraculously achieve this, they actually put they get that back. How do they do that? Like, they could just ligate it together and you have this deletion, but repair enzymes can sometimes repair it so that it doesn't achieve what I just said, a deletion. So if you delete this and it stays gone, okay, fine. You're missing AGT. It's gonna go right from proline to tryptophan. That's fine. But in truth, you actually do have some ability to repair these. Not all of them. Sometimes you can repair DNA. How did he know to put AGT there? Like, did he go to class and realize, well, that protein has an arginine at position number 3. Remember, these enzymes are not too smart. Every time I ask you, how did they know? You always say, they didn't know. They're not that smart, and they did something with trial and error. So he must have had there must be some DNA around here that he's just copying off of, right? Like, does he have a friend he can copy from? Like, let's say this is chromosome number 4, and there's a gene on chromosome number 4. Does he have a friend? Like, is there like a backup copy of chromosome number 4?
Yes.
You have backup copies of chromosomes? He's right. There's a backup copy of every single chromosome. The one that you got from your other parent. Like, let's just say, for argument's sake, the one I've drawn up here is the one that's from the mom. The dad's backup copy is there, and that's exactly what you need him for. Once in a while, you have a double-stranded break, and you lost some DNA, and the repair enzymes are like, there's no way I can just guess 3 random amino acids. Sorry, 3 random bases to make 1 amino acid. So what he does is he says, let me find the other strand, the other chromosome, which is chromosome 4 from the other parent. What do you call the other chromosome? Like, I'm calling it the other, but officially in genetics class, what do you call the other chromosome? So, you got your chromosome 4, and the other one's called starts with homo. It's not the identical chromosome, it's homologous. It's similar because your dad's chromosome number 4 probably has the identical sequence for any given gene, probably. But everybody knows mom and dad's DNA is not identical, but at least there'll be some reasonable stretch of DNA here that would give you a reasonable functional protein. Like, if mom has an arginine here, let's just say for argument's sake that dad's got a different 3 letters there. What 3 letters do you think dad might have if it's different? It's different, but it's still functional. What 3 letters do you think dad might have?
AGA.
He says AGA. If it's true that you have a whole other chromosome and it's got AGA and it's going to be dad, we're going to draw it in a different color. So now I'm just going to leave it because this is the other chromosome. This is a homologous chromosome. And he thinks that maybe in the critical region here, the dad has AGA. A instead of AGT, which would still code for arginine, which is fine. And if that's true, you would call that a polymorphism. So here's the difference. That T in mom is replaced with that A in dad. Here's the codon that we're asking about. That's a difference. Poly means more than one, so polymorph, shape, so there's a different actual amino acid. So polymorphism, it's a glorified word that just means a single letter difference. Usually it's a single letter difference in the base. A single, NT means nucleotide difference in the sequence. It's also possible that dad could have a whole nother amino acid there. Instead of this plus charged amino acid, do we have any other plus charged amino acids that are possible?
Lysine.
Yeah. And so lysine would just be AA, and possibly AAG or AAA. You have to change it to AAG or AAA. But let's just say for argument's sake, dad, in one case he has a single letter polymorphism, but that change does not change the actual amino acid. You could change the DNA. You could have 4 different polymorphisms. You could also have A, G, C. That's still gonna code for arginine, because arginine has 4 possible codons. You don't have to have AGT. You can have AGU, AGC, all those 4 plus 2 more. There's 2 more that code for arginine. Arginine's got 6 codons, but all of those this particular polymorphism of changing the 3rd letter, it still keeps you as arginine. But that polymorphism doesn't make you behave differently, or doesn't make your whatever protein this is. But you could potentially change this to lysine. And maybe lysine has a slightly different 3-dimensional shape, and the protein works a little differently, and maybe inheriting that makes a difference. But the bottom line is, even if you have a difference, it's not that big of a deal. Dad's version of this chromosome is still functional. All right, so I'm gonna go back to, let's assume that this particular stretch of DNA is completely identical in both versions of chromosome 4. So there is not a polymorphism. Mom's stretch of 9 nucleotides and dad's stretch of 10 nucleotides that you inherited are identical. Some other places on their chromosomes they could be different. And if this is what you had, then you could have a protein that says, I don't know what we lost here, but let me go copy off the other chromosome. That's basically what it is. So here's the money question. How does this protein, the DNA, it does deletion mutations, or it does, when you've lost sequence, and this is completely gone, and you're trying to fix it, how does that protein know? And the simple answer is, it copies off of the other chromosome. It grabs the homologous other chromosome, and of course you know it's not gonna say, hey, can I just look at you and then copy it? They're not that smart. You know what's gonna happen. Which one of you can tell me exactly how a protein is going to have to function that's trying to attach something to CCC? So if you ask the polymerase, how do you know to put the next letter? Didn't we say the next letter was supposed to be A to G and a T, right? There's going to be a protein, a polymerase. It's going to come in here and it's going to put an A, a G, and a T. And how is it going to do that? Based off of what base pairing? Point to up here is the actual basis he'll be grabbing the base pairing with by trial and error. You're right about that. It's trial and error. But he's trial and error base pairing with whom? He's base pairing with that, this. Which strand is he grabbing a hold of and saying, all right, I'm copying off of, taking the complement of you?
The bottom.
So he said the bottom homologous strand. I'm going to put it in bold, the ones that he really cares about. See these 3 here? He wants to copy by using those 3 letters as his guide. So what that means is, these G's are in the way, and the reason why I say that is, what we're gonna do is we're gonna chew those away. So the repair enzymes have to do one thing first. They have to have a little chopper that eats away these G's. I'm just gonna put, like, one of them's already gone. It's chopping these G's up. So step one in causing this problem to be resolved is you have to cause these triple C's to get stuck down here. See these three G's here? They're normally nicely paired with those C's. And what's gonna happen is they're gonna get a new girlfriend. This is gonna be your new girlfriend. I'm gonna give her a little heart. She's gonna say, watch me cheat on these little guys. These triple G's are gone because he got rid of them. These three C's are gonna come down here and say, hey, how about you guys leave? I wanna come in here and base pair. So what that means is, if you have single-stranded DNA that's part of a damage, if it could speak, it'd say, look, I've been damaged. I'm gonna come in here and kick you guys out and base pair so that I can allow my polymerase to read off this tCNA. So we basically have to push all those C's and this AGT, we have to push them up and make a bubble. So I'm gonna actually draw that over here. I'm gonna redraw that whole thing as a bubble so you can see what we're talking about. So this C, it was the original part, because you can say, hey, who's butting in on my, I used to be paired up. This was my pair. And the thing that snuck in was this. This used to be damaged, and it snuck in. It was a piece of single-stranded. The reason why it's single-stranded is because this Pac-Man came in and ate away the bottom G's. So this is the original strand of C's, and it continues. A, G, T, T, G. And we basically made a big old bubble. And here's the critical basis that we really need to read off of. We want the polymerase to be sitting across from these and doing trial and error. And if you ask him, how did he get there? Well, he had to go down to the partner, the homologous strand, pull it apart. And the way you could say is that this little heart says, hey, can I be the girlfriend for a while? Could you please leave? So the original girlfriend has to wait for a while, and he has a new girlfriend for a while. And then he does the critical thing, draw pink. He puts he does these three bases by trial and error. And he actually goes a little further, but that's basically where he gets these bases. And the important thing is, this is temporary. This bond is never, this is the blue strand here, it stays covalently bound to its partner. And I don't know if this is woke or not for me to say this, but like, she pairs up with her original partner again. Maybe she should leave him forever, but like, what's gonna happen is the red guy's gonna leave, right? The scarlet girl's gonna leave, and then we're gonna zip this back up. And if the scarlet one could speak, it would say, thank you for letting me temporarily come in and create that. I'll just redraw. Let's be good. She leaves quickly. And then in pink, she puts a big old AGT. So if you ask, how did you make the AGT? This process in biochemistry textbooks, they write it as invasion of the strand. And that is kind of what happened. This triple C used to be part of the damaged strand, and it actually poked in and said, hey, would you guys separate for a while? I need to read off of this TCA. And this bubble, where you're kind of separating, some people call it a D loop. And it's only temporary. Like these letters that these red and pink letters, as they're made, they don't stay jammed in there. They eventually spit out, and the blue guys zip back up. So the original homologous strand goes back together, and after a period of time, the polymerase says, right, that's enough. I think I have enough to make up for what I was missing. The green marker is still gone. So that's called double, and the reason why I went through the trouble of telling you that is, A, you're gonna be able to answer the question, how does double-stranded breaks with loss of nucleotides work? If you have a double-stranded break and you didn't lose any DNA, just ligate. DNA ligase is enough if it's a clean cut, but if you mangle a bunch of the bases and you've lost 3 bases, basically you have to take your double-stranded DNA on one side, trim it a little bit so that it can invade. And it invades the lower strand, temporarily pulls it apart, and if it could speak, it would say, sorry, I've just come over here to copy off of your bottom strand. And then I'll leave. That's how you can kind of put it in regular language. And that's what I'm looking for on the exam. I'll ask, how does this process work? And I want you to break it into 3 steps. Step 1, you must make the damaged DNA, you gotta chew it up, because I wrote these as red eventually, but this double-stranded DNA can't do the invasion. You have to get the blue DNase. That's what this is. DNase. There's a DNase function as a repair enzyme that eats away. I call it they actually call this nuclease. Nuclease. And it cuts the DNA. And once you make some single-stranded overhang, what's the number on this? Is this a 5 or a 3 prime at the end of that third C, right by the heart?
5.
He says it's 5. It's actually a 3 because the direction is always 5 to 3 going that way. And the lower strand, I'm going to draw that in blue. It's going in this direction, and it's 5. Like that C would have a 5 all the way, and this G here would have a 3. So this C here would have a 5. So the arrow on the bottom strand's coming towards me, the arrow on the top strand's going away from me. So the first of those 3 C's is a 5. So that's a 5 on this side, it's a 3 on that side. So you need a 3 prime end. This is a 3 prime end that's invading the double-stranded DNA, making a bubble and reading off of the lower strand by trial and error. So that's the general idea of that. And that's not only helpful for understanding how do you repair DNA, but that's the essence of how they fix broken DNA using CRISPR. So in CRISPR, Cas-CRISPR, which is this miraculous thing that they won the Nobel Prize for recently, it describes the process by which you could take DNA out, take a cell out of somebody, and find a mistake in their DNA and fix the mistake. And that's what CRISPR is. SPR. And CRISPR, we described a little bit in lab, but just as a brief summary, CRISPR was stolen from bacteria. Here's this little flagella. This is a bacteria. And viruses used to invade these bacteria here's little viruses. That's actually called a phage, P-H-A-G-E. Phage is a virus that infects bacteria. And the bacteria would get sick of being infected over and over again by the same phage, and so they would fight back. And so this must be the phage that doesn't murder the bacteria, it keeps it alive. And the phage would come in, and the first thing the phage would do is say, here's my sequence, A, C, C, G, G, whatever. Please make that into my daughter's proteins. So viruses do the same thing in bacteria they do in us. They shove DNA or RNA into us and go, make this. And our cells are very accommodating and very, very helpful. They're okay, whatever you say. My ribosome will make that. So bacteria have little ribosomes, and they'll say, I'll turn that into RNA and make you some messenger RNA that's viral, and I'll make some viral daughters. And that whole process creates a bunch of daughters. But eventually the bacteria evolved to where they could fight back against this. And what they would do, they can't stop the first guy from coming, but they would create this. You see that sequence right there? Anybody recognize why I did it like that? Here, I'll even put the numbers. It's going in that direction. Anybody recognize that sequence?
It's complementary.
He says it's complementary. That's what you would call an attack sequence. It's attacking this. Now it's too late, this bacteria has already been invaded, but she's planning ahead. If she doesn't die and she creates daughters, she's going to bequeath that to her daughters. She's going to let her daughters have this little sequence, and if the daughter could speak and say, hey mom, why did you give me that? It's not part of your original DNA. Let's imagine that the daughter was smart enough to spy on the mom's DNA. And she would say, oh yeah, what had happened is we keep getting invaded. I created some new DNA. It's gonna cause you to have this attack sequence, so if that virus's friends ever come after you, you can just produce this DNA. This DNA will stick to their DNA, make it to where it can produce. This would work with RNA too. But bottom line is, this is the next best thing to an antibody. This is a little bit like an antibody, but with inside the cell. Instead of protein to protein, it's DNA strand to DNA strand. And if this virus doesn't kill the original host, maybe some months later, the virus is long gone, but another virus comes along with that same sequence. This bacteria is like, aha, I can feel that virus is infecting me. Let me turn on the DNA that produces this attack sequence, and it would bind. And so this is what it would look like. It would look like this, and we were mentioning this before. If you're trying to cause that sequence to be turned into messenger RNA, or if it is messenger RNA, these would be used. But if you're trying to get it into protein, it's bad to have it be double-stranded, and it's bad, furthermore, that once it's double-stranded, there are special proteins who like to eat it. These might be nucleases, and the nucleases like to eat, particularly double-stranded RNA. In the case of double-stranded DNA, it might interfere with the production of messenger RNA. The attack sequence, technically the attack sequence is supposed to be all in the RNA field, but the principle is the same. If you make this stuff double-stranded, the nucleases will eat it. If they eat it, the virus will be frustrated and say, hey, how come I didn't get any daughters? And this bacteria will say, well, because your cousins from a couple months ago, they came in and I remembered the sequence. And I made the opposite strand, and my opposite strand is gonna attack your invading strand and neutralize it. So that process was stolen, and when they originally discovered these sequences in the bacteria, scientists were like, well, what gene is that? What protein is that? It doesn't code for a protein. And so they argued about what it was, and they finally said that they're clustered, because every time you get an infection, you create another one of these, so there's a certain database of all the bacteria that have ever infected a given bacteria, all the viruses. So they're located in this cluster. So they're clustered, regularly interspersed. So when you look at the DNA of a bacteria, you will find this region, and there's this little cluster of attack sequences in their DNA. Here's bacteria, sorry, virus A, phage virus A. Here's phage virus B. Here's phage virus C. D, etc. So all the places where they store these attack sequences were finally identified, and the scientists were like, what's the purpose of this sequence? They didn't understand it yet. And if I'd named it, I would say this is your bacterial attack database, or I would call it like your immune database, something like that. That would tell you it's for attacking invading viruses. It's the little sequences that you stole from one after another different virus. Every time they invaded, you kept a little bit of their sequence so that you could make an opposite strand so it would attack. Clustered, regularly interspersed repeats. So interspersed is IS. Clustered, regularly interspersed. Palindromic. Palindromic just means that they read forwards and backwards the same and repeats. So the reason why we use this is sometimes a person has a problem in his DNA and you have to find the right spot. And I'm not going to tell you about it today, but at least you will learn whenever we do start talking about it. When I say CRISPR, you're going to know that it was this attack sequence stolen from bacteria. And we use it in laboratory settings to help repair broken DNA. So you should know where it started from. So by the time you're done today, you should be able to at least describe how you fix broken DNA that's not just broken, but it was broken and we lost. And then the second thing is about CRISPR, and that is all I have for today.